Hello dear students. Today we are going to study a very important chapter of class 12 biology, that is biotechnology principles and processes. We are also going to attempt previous year questions from NEET in this video. Okay. Now students, this chapter is extremely important for both NEET and class 12 board examinations as it carries a high ratio of 7.5% of the total marks in NEET exam. Okay, also this chapter will act as the base for the next chapter, okay, which is biotechnology and its applications. So you must read this chapter thoroughly. Now, students, an interesting fact about this chapter is that in this chapter, we will be able to divide. What is this? This is DNA, right? This is DNA. So, we will be able to divide DNA into small fragments. Okay. For example, I cut the DNA from here. This is a small fragment and combine these small fragments with other small fragments to produce a new DNA. Okay. Now students, you must be wondering, why are we producing a new DNA? Right. I will answer this question further in this video. This means that the students who wish to pursue biotechnology as their field must watch this video to understand the concepts thoroughly. Okay, now let us start with the chapter. Alright, now let us understand what is biotechnology. Now, according to the European Federation of Biotechnology, that is EFB, they have given a definition of biotechnology. Right, now this definition encompasses, that means it comprises of both traditional view and modern molecular biotechnology. Okay, so what is the definition? The integration, that means coming together or joining, right? So the integration of what? The integration of natural science and organisms, cells, parts thereof, that means parts of the cells and organisms, and molecular analogies for products and services. Now they're saying that what is biotechnology? Biotechnology is the integration of natural science with organisms, cells, parts of these cells, molecular analogies theories for products and services. Now, in simple words, what is biotechnology? Biotechnology deals with the techniques of using organisms or enzymes from organisms to produce products and services useful to humans. Now, let me explain this simple definition to you. Students, biotechnology deals with the techniques. Major keyword. What is biotechnology? It deals with the techniques of what? Of using organisms or enzymes. Okay, we are using organisms and enzymes that are extracted from organisms. For what purpose? To produce products and services that are useful to humans. Clear? Alright students, now that we are clear with the definition of biotechnology, let us understand the principles of biotechnology. Now there are major two principles of biotechnology. Okay, the two core techniques that enable the birth of modern biotechnology are genetic engineering and bioprocess engineering. Now let us study them in detail. What is genetic engineering? the techniques to alter. Alter means to modify, right? To alter the chemistry of genetic material. What is genetic material? DNA or RNA. To introduce these into host organisms. What are host organisms? Another organism in which a DNA or a genetic material is introduced. Okay, and thus change the phenotype. What is phenotype? Physical appearance of an organism. Okay, phenotype of the host organism. Now, in simpler words, what is genetic engineering? Genetic engineering is the technique to modify the DNA and introduce that modified DNA into another organism, which is known as the host organism, to change the physical appearance of that host organism. Clear? Now, what is bioprocess engineering? It is the maintenance of sterile. Sterile means microbial contamination free. That means germ free, right? Now, it is the maintenance of sterile ambience. What is ambience? Environment. In chemical engineering or genetic engineering, same meaning. Chemical engineering processes to enable growth. Enable, that means we're allowing the growth of what? Only desired microbes or eukaryotic cells in large quantities for the manufacture or production of the biotechnological products like antibiotics, vaccines, enzymes, etc. In simpler words, what is bioprocess engineering? It is the maintenance of germ free environment to produce desired, only desired microbes to further produce biotechnological products. Okay, this is what is bioprocess engineering. It is the maintenance of germ-free environment. Now, in this germ-free environment, what will happen? Genetic engineering will happen, right? Modification of the DNA will happen, and the desired microbes will be grown. Only the desired microbes, right? That is why it is germ free or microbial contamination free, so that there is no contamination and only desired microbes, no extra microbes are grown. Okay, why are these microbes grown? The question is, why are these microbes grown? They are grown to further produce biotechnological products. For example, antibiotics, vaccines, and enzymes. Remember that these antibiotics, vaccines, and enzymes are produced from these microbes or eukaryotic cells. That is why we are allowing them to grow in this environment. Clear? Now, these were the two principles of biotechnology. Okay, now that we're clear with the definitions of two major keywords of the chapter, that are biotechnology and principles of biotechnology, let us now talk about the processes, of biotechnology. Okay, starting with how are genetically modified organisms created. Now, students, what are genetically modified organisms or GMOs? What are these? Genetically modified organisms are the organisms that are genetically modified or whose DNA has been altered or modified, as we studied before, with the help of biotechnology. Right? Those organisms whose DNA has been altered with the help of biotechnology are known as genetically modified organisms. Clear? Now we are going to understand how this DNA is altered with the biotechnology. There are three basic steps in genetically modifying an organism. Okay. The first step is identification of DNA with desirable genes. Firstly, we need to identify what genes are desired by us. Okay, then only we will extract out those genes, right? Second is the introduction of identified DNA into the host. Now we have selected desired genes. Okay, and now we are going to introduce these selected desired genes into the host. Then the third step is to maintain the introduced DNA in the host and then transfer of that DNA to the progeny. Okay, genetically modified organisms are the goal to evolution of GMOs. Okay, they are the goal to evolution of that genetically modified technology into the progeny. That means that we not only have to create that genetically modified organism for one generation, but we have to pass on that genetically modified organism technology into the further progeny. Right. So once it is introduced in the host, once the desired gene is introduced in the host, then we have to maintain that desired gene or that introduced DNA in the host and transfer, and we have to enable the transfer of that DNA to into the progeny. That means we have to put a check that that DNA is being transferred to the progeny, to the further generation as well. For example, if we are introducing a gene in the F1 generation, we have to make sure that that gene is being passed on to the F2 generation. Clear? Now students, we know that these GMOs or genetically modified organisms have been genetically altered. Right. Now remember, in the starting of the video, I told you that we are going to create a new DNA. Right. That is what we're doing here. We are genetically modifying the already present DNA and creating a new DNA by introducing it into the host. Right. But what is this new DNA called? This new DNA is called recombinant DNA. Why is it called recombinant DNA? Because we are recombining this DNA with the old DNA. We are recombining our desired genes with the old DNA to produce a new DNA. That is why it is known as recombinant DNA. I hope this is clear. Okay. Moving further with what are the tools of DNA technology. Now there are various tools of recombinant DNA technology and they are majorly categorized into restriction enzymes, cloning vectors, competent host and DNA transfer into host tools. These are the tools that I will list only in this video, which is provided by EduRef. These are the major classifications simplifying what is written in your NCRT. Clear? Okay, before discussing these tools in detail, let us first look at an experiment concerning how was the first recombinant DNA formed. Okay, now we're going to see an experiment. It is going to be very interesting. Now, the construction of the first recombinant DNA emerged from the possibility of linking a gene that encoded means that had a code that worked for antibiotic resistance. Okay, now how did they start? They got to know that they can link a gene. Okay, they can link genes together, and they selected a gene that coded for antibiotic resistance. Okay, now, and they had a native cloning vector. Native means it was already present. An organic or native cloning vector. Cloning vector. What is a cloning vector? That allows that grants another gene to be added up in that vector. Okay, the vector will delete one of its gene and will add up the gene. Okay, but it will not do it on its own. We have to cut out the gene from the vector, and we have to cut out the gene from another organism, and we have to paste this gene in this vector where we have emptied up the space. Right. Now, what is a cloning vector? It is another tool. It is a tool of the DNA technology that we will study further in detail. For now, that cloning vector was known as plasmid. Okay. Now, students, there were two scientists. There were two scientists, namely Stanley Cohen and Herbert Boyer. Now, what did these two scientists do? They formed the first recombinant DNA in the year 1972. How? They selected a gene that coded for antibiotic resistance. Okay, that gene was present in another organism. So they cut out that gene from that organism and they selected a cloning vector. A cloning vector that was already that was an organic vector for the gene that was already linked up with that gene. Okay, this gene used to work with this cloning vector before they discovered it. Okay, now they discovered that there is a cloning vector that works with this gene, and that cloning vector is plasmid. Okay, so what did they do? They deleted one of the gene from the cloning vector that conferred, that granted permission for this gene to come here and get pasted in the vector. Okay, so they deleted one gene from the vector and they cut out one gene from the organism that coded for antibiotic resistance. Clear? We have one gene in one organism and we have the cloning vector. So this gene here, for example, this is a gene. This gene here encodes for antibiotic resistance. So they cut this gene out and they cut one gene out here. Okay, for the location of this gene to be pasted. Okay, and they pasted this gene here in this plasmid. Now, this new DNA strand is introduced in the host organism. Okay, and this changed the phenotype of the host organism. Clear? This is what they did. I hope this is clear. Okay, but the question arises, how did they how did they cut the DNA? We are saying that we cut one gene from this organism and we cut one gene from the vector for the gene to be pasted here. Remember that a gene will be pasted on a specific location. You cannot just delete any gene from the vector. Okay, there will be one gene in the vector that will confer or that will grant the permission to the foreign gene to be pasted here. Okay, not any gene can be deleted from the vector. Okay, so what are they doing? They're basically cutting the genes. But how are we cutting the genes? If you remember, in the very beginning of the video, I said that we will cut the genes, we will cut the DNA into small fragments, we will combine those fragments and we will produce the new DNA. What is the new DNA? Recombinant DNA, right? But how are we cutting the DNA? We are cutting the DNA with the help of molecular scissors. Okay, molecular scissors is another tool that we are going to study. Okay, but we will study this later on. Now, we know that we have tools in the recombinant DNA technology. Okay, let us study these tools in detail along with the molecular scissors. You will get to know what is molecular scissors further on. Now, let's talk about restriction enzymes first. Okay, so as I said before, these enzymes are also known as molecular scissors. What are molecular scissors? Restriction enzymes. Okay. Why? Because they cut both the foreign and vector DNA into fragments to form the recombinant DNA. What did they do? They cut both vector and foreign DNA to into fragments to form recombinant DNA. Clear? As you can see here, this is an example. Okay, this is an example. They're not actually cutting the DNA with the scissors, but they're representing restriction enzymes here. Okay, restriction enzymes are enzymes that cut the DNA. We don't actually use the scissors, but we use restriction enzymes. Okay, this is just an image used to represent restriction enzymes. Now, let us discuss an interesting fact about these restriction enzymes. Yeah, okay. So, the first restriction endonuclease. We're going to talk about the first restriction endonuclease. What was it? It was HIND2. This is the name of the first restriction endonuclease whose functioning depended on specific DNA nucleotide sequence. Okay, and it was isolated and characterized 5 years later. Okay, it was found that HIN2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence for HIN2. Now, what does this mean? They are saying that there was the first restriction endonuclease or first restriction enzyme. Endonuclease is a restriction enzyme. Okay, now we're talking about the first. We're talking about the first restriction endonuclease, which was known as HIND2. This is the name of the endonuclease. They are saying that this endonuclease has a property that it always cuts. This restriction enzyme has a property that it always cuts DNA at specific locations. That means it does not cut DNA anywhere, just anywhere. That is what I said before, right? You cannot just cut the gene out of the vector anywhere, right? You will only cut it in the conferring positions or in the specific locations. So how was this find out? How was this found out? This was found out by the help of HIND2. HIND2 associated this theory. Okay, it credited that DNA can be cut only on particular points, only on specific locations. Okay, scientists figured out that HIN2 always cuts the DNA by recognizing a sequence, and that sequence was of 6 base pairs. Now we know what are base pairs, right? So they're saying that HIN2 recognizes 6 base pairs and then cuts the DNA at that location. Okay, now this 6 base pair sequence is known as the recognition sequence. Clear? Now students, we know what restriction enzymes are, right? But it is also important to know to what category these restriction enzymes belong to. So, restriction enzymes belong to a larger class of enzymes called nucleases. Now, restriction enzymes belong to a higher class which is known as nucleases. Nucleases are also enzymes. Okay, and these nucleases are of two kinds, exonucleases and endonucleases. Now, students, we have a brief idea about endonucleases. We studied about endonucleases when we discussed HIN2, right? When we discussed HIN2, we studied that HIN2 is a endonuclease or a restriction enzyme. But now we'll study them in detail, what they mean. Now, exonucleases remove nucleotides from the ends of the DNA. Ends of the DNA. Okay, whereas endonucleases make cuts at specific positions within the DNA. You understand the difference? We know that the restriction enzymes remove nucleotides, right? Now, exonucleases are a kind of restriction enzymes that remove nucleotides from the end of the DNA. Exo means end, okay, exit, whereas endo means inside. So, endonucleases make cuts at specific positions that are within the DNA, inside the DNA. Okay, so if this is a DNA strand, then an exonuclease will remove nucleotides from the ends of the DNA, whereas an endonuclease will remove the nucleotides from the center or within the DNA. Got it? This is endonuclease and this is exonuclease. Clear? Okay. Now, each restriction endonuclease functions by inspecting the length of the DNA sequence. Endonuclease checks the length of the DNA sequence first. Once it finds its specific recognition sequence. Now, we know for exonuclease it is very easy. It will just cut the ends of the DNA, right? But for endonuclease, it is a hard task. Endonuclease first has to inspect the length of the whole DNA, right? Then it has to find the recognition sequence to which it will bind to the DNA and cut the gene of the DNA. Okay, first it will find the recognition sequence that matches with its DNA and then it will bind to that DNA. It will bind to that DNA and then it will cut each of the two strands. We know that the DNA has two strands. It is a double helix structure, right? So there are two strands of the DNA, right? So it will cut both of the two strands of the DNA of the double helix structure at specific points in their sugar phosphate backbones. We know that DNA consists of nucleotides and sugar phosphates. So it will cut at specific points in their sugar phosphate backbones. Now, each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Now, we know that it recognizes a recognition sequence, and that recognition sequence is known as palindromic sequence. That recognition sequence is palindromic. But what is palindromic? We will study what are palindromes later on. But firstly, we will attempt a previous year question from NEET, that is from the year 2020. Okay, now in this question, they are asking about palindromic sequence. Okay, we have not studied this yet. We will study it later on in this video. But whatever we have studied about palindromic sequence, we are going to attempt that question on the basis of this. Okay, this question is on the basis of what I told you here. What I told you here in the last line. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Okay, that means that each endonuclease recognizes the specific recognition sequence which is known as the palindromic nucleotide sequence. Okay, and that restriction endonuclease cuts this palindromic nucleotide sequence not in the center, just little away from the center. For example, this is the palindromic sequence. This is the recognition sequence which is known as palindromic nucleotide sequence. So the restriction endonuclease will cut this palindromic nucleotide sequence a little away from the center. This is the center. It will cut it here, a little away from the center. Okay. Now we're going to attempt this NEET previous year question on the basis of our understanding. Now, given below are two statements. Statement 1. Restriction endonucleases recognize specific sequence to cut DNA known as palindromic nucleotide sequence. Correct. This is correct. Right? We know that restriction endonucleases recognize a specific sequence which is known as recognition sequence. Okay, and that recognition sequence in turn is known as palindromic nucleotide sequence. So this statement is correct. Next statement. Restriction endonucleases cut the DNA strand a little away from the center of the palindromic sequence site. Now this is what I told you just now here, right? That they cut the palindromic nucleotide sequence a little away from the center. Okay, this is what they're saying in statement 2. So this is also correct. Right. Now, in the light of the above statements, choose the best suitable answer from the options below. Both the statement 1 and 2 are incorrect. No. Statement 1 is correct, but statement 2 is incorrect. No. Statement 2 is correct, but statement 1 is incorrect. No. Both the statement 1 and 2 are correct. Yes. So the answer to this previous year question is D. Understood? Now students, as I said before, we will discuss palindromes in detail. Let us study them. Do you know what palindromes are? These are group of letters that are read the same way in forward and backward direction. Okay, what are palindromes? Palindromes are group of letters that are read the same way in forward and backward direction. Okay. For example, consider this word. Malayalam. Now, this word reads the same in forward and in backward direction. Malayalam and Malayalam. Malayalam. Malayam. Okay, so it is what? It is a palindrome. Clear? Now, against a word palindrome where the same word is read in both directions, the palindrome in DNA sequence is a sequence of base pairs that reads the same on the two strands when orientation of the reading is kept the same. Okay, so in DNA sequences, what is palindrome? In DNA sequences, palindrome is a sequence of base pairs. Okay, now these base pairs are on the two strands, strand 1 and strand 2 of the DNA. Okay, and these palindromic sequences are read the same way when the orientation of the reading is kept the same. For example, GAATTC 5:3. Now 3:5 would be CTTAAG. Got it? Now if I read it this way, it is GAATTC and GAATTC. Clear? These sequences are what? These are palindromic sequences. Clear? Alright students, now let us attempt a previous year question from NEET 2022 on palindromic base sequences. Now, in the following palindromic base sequences of DNA, which one can be cut easily by particular restriction enzyme? Now, a restriction enzyme will only cut the palindromic base sequence. Right. Let's see which one of these is a palindromic base sequence. GAATTC. When read this side, GAATTC. Okay, this is a palindromic base sequence. Let's check option B, CTCAGT. Okay, ACT. No, this cannot be a palindromic base sequence because it should have begun with C. Right, now option C, GTATTC. G, correct. AA, no. After G, there should be T, not A. So again, this is wrong. Option D, GATACT. Okay. AG, no. Again, this should have begun with G, not A. So this is also not a palindromic base sequence. That means the answer to this previous year NEET question is option E. Clear? Okay, students, now we're going to study a very interesting topic of this chapter, which is separation and isolation of DNA fragments. Now we have fragmented the DNA using the molecular scissors, using the restriction endonucleases and restriction exonucleases. Right, now what will happen next? We will separate these fragments and isolate these fragments to combine them with other fragments and form a new DNA. Right, so we are on step 2 now. The cutting of DNA by restriction endonucleases results in the fragments of DNA. That is what I told you. These fragments can be separated by a technique known as gel electrophoresis. Now this is very important and you will have a question in your exam from this topic for sure. Okay, so let us see what this is about. Now, since DNA fragments are negatively charged, we know that DNA is negatively charged, right? So since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode, right? The anode is what? Anode is positively charged, right? We are going to carry out the electrophoresis of the DNA. Electrophoresis means we are going to involve electricity in this process. So we are going to have a cathode and we are going to have a anode, right? So anode is positive. That means the DNA fragments will rush towards the anode, right? Okay. Now, how will that happen? They will rush towards the anode under an electric field through a medium or a matrix. They need to be in a medium, right? We need to have a medium in which these DNA fragments will be present, and we need to have an anode and a cathode, right? So what will be that medium? Nowadays, the most commonly used medium or matrix is agarose gel. That is why it is known as gel electrophoresis, because the medium that we use here is agarose gel and we carry out the electrophoresis of this DNA fragment. That means it is known as that's why it is known as gel electrophoresis. Clear? Now, this agarose gel is extracted from the seaweeds. It is extracted from the seaweeds. These are the 1-mark questions that can come in your exam. Okay, this agarose gel is extracted from the seaweeds. Okay, and it is a natural polymer. What is it? It is a natural polymer. Clear? Now, the DNA fragments separate or resolve according to their size through the sieving effect provided by the agarose gel. Now, we know that the smallest fragment will rush faster towards the anode. Here is the anode, here is the cathode. Okay, so the smallest fragment will rush faster towards the anode, and then the bigger one, then the bigger one, then the bigger one. So these fragments arrange them according to their sizes through the sieving effect. What effect? Through the sieving effect. Okay. Through the sieving effect, these fragments arrange themselves according to their sizes. Right. Now this effect is provided by what? Provided by the agarose gel. Okay. Now, hence the smaller the fragment size, the farther it moves. That is what I told you. The smaller the fragment size, the farther it moves. Is that clear? Now, students, this sieving effect cannot be seen by a human eye. Okay, we cannot see this effect. So we have to stain these fragments. We have to color these fragments for us to see them. Okay, so we use a stain. We use a chemical which is known as ethidium bromide. Okay, we stain these fragments with ethidium bromide and then we provide UV radiation. We provide UV radiation on this area, okay, on this gel. And what happens? Then we can see the sieving effect provided by the agarose gel. Okay, then we can see that these DNA fragments have separated and isolated themselves according to their sizes. Clear? Okay, now let us attempt a previous year question of NEET on the basis of gel electrophoresis. Now, DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appear as. Now, this is something that I did not tell you. Okay, we are going to attempt it in the previous year question. They are saying that when we stain these fragments with ethidium bromide and then we provide UV radiation to these fragments, what color do they depict? So, students, they depict the orange color. Okay, they depict the orange color. So, from all of these options, option D is the right answer. Option D is the right answer. They are of bright orange color. Okay. Okay. Coming on to the last topic of this video, which is cloning vectors. Now, as we discussed before, the tools of biotechnology are restriction enzymes, cloning vectors, and etc. Right. So, we are coming to the cloning vectors. Now, what are cloning vectors? Cloning vectors are vehicles that transport recombinant DNA into the competent host. Clear? They transfer the recombinant DNA into the competent host. But how do they do that? These cloning vectors add up the desired gene in their own genetic material and then transfer the new DNA into the competent host. Clear? Now, the plasmid is a cloning vector. Okay, as we discussed before, plasmid is a cloning vector. Now, the plasmid is the autonomously. That means it replicates on its own. On its own. Autonomously replicating circular. What is the shape? Circular extra chromosomal. That means it has extra chromosomes DNA that is used as a vector to transfer the new genome in the host. So what is a plasmid? Plasmid is the autonomously replicating circular extra chromosomal DNA that is used as a vector to transfer the new genome in the host. Okay, so the vehicle that we're talking about here is plasmid. This is the diagram. Now this is plasmid, okay, which deleted its own gene and added up the new desired gene. Okay, this is the new gene. Okay, that is being added up here. And when it was combined with the plasmid, it produced the recombinant DNA. Now this recombinant DNA is added up in the host organism. Clear? Now in the next video, we will talk about the features that are required to facilitate cloning into a vector. Okay? Okay students, I hope you people have now understood what we discussed in this video. To know more about this topic, you can join the EduRiff Infinity Plan, log into your Class 12 Biology section, check out the chapter Biotechnology Principles and Processes, unlock the locked videos, documents and test series in less than 80 rupees a month. Thank you.